Good afternoon, everybody. I'd like to thank Julio for inviting me to participate in this meeting and really to bring us all together to have this conversation about this really important subject. So the task at hand for me is to talk a little bit about the research that's planned and underway that the NIH is currently supporting. And a fair amount of what you will see, you've already seen today because WAFA covered a fair number of the studies that I'm going to at least touch on. So I can save a little bit of time by not necessarily skipping over them, but giving us, just reinforcing the messages you've already heard. So again, what we're talking about in the spectrum of HIV prevention is a whole range of activities from dealing with behavioral, structural, and vaccine-based approaches, adult male circumcision to deal with the earliest events, to PrEP and microbicides at the time of exposure, all the way through to treatment as prevention, which is the primary focus of the meeting today. So I'm going to start by going back through this and starting with a little bit about topical microbicides, because I think actually this matters a little bit for treatment as prevention, because both PrEP and microbicides need to be considered in the context of using ART as prevention. So in terms of our future directions for topical microbicides, purely from a research perspective at this point, so we really absolutely need to complete the phase 2b trial, the VOICE trial, because we really need to generate the evidence or not that moves tenofovir gel toward licensure. And VOICE will provide a significant amount of data on the safety of tenofovir gel because it's a daily use trial. And then the combination of VOICE plus Caprisa plus the FACTS trial will give us the data we need to license the gel. Then we have to worry about manufacture and moving this into a real-world product that's available at a cost that is sustainable. At the same time, we need to continue development of rectal microbicides, and ultimately we need to move quickly to second-generation products that are less behaviorally sensitive, I guess is the nicest way to say it, that basically you don't have to worry about remembering to use every day or is in some way coitally dependent, and you actually have to have that recall at the time of passion that, oh, hold on a minute, it's the same problems we have with condoms. Ultimately, we do need to be able to evaluate vaccines and microbicides together and move toward novel clinical trials methodologies. So just to remind everybody, VOICE does examine both vaginal and oral interventions. My point about showing this slide is there's been some mention of FemPrep. It's probably mainly in the hallways. It's like, what's going on with FemPrep? I think the point, though, is that VOICE is exceedingly well positioned to either confirm the FemPrep result, think about what that would mean, that would mean p-value of 0.5, or refute it. And so as these studies move toward interim analysis by DSMB and or toward completion, I think we will get a very firm response one way or the other out of VOICE, because this study is very well powered to answer the question. And we may see that the problem was purely an adherence issue. We may also see that there was actually some biology going on. And in fact, maybe these drugs orally do not work in women the way they work in men. Needless to say, we also have the topical arm, which, unless something truly untoward happens, should continue toward completion. At the same time, we have moved the PrEP study, as WAFA mentioned today, to IPREX-Olay. And I think that was one of the clever marketing schemes that has been developed lately. In terms of the next generation of PrEP, clearly VOICE also helps answer this PrEP question, but it's time for a research perspective to also start moving toward longer acting formulation, whether we're dealing with nano formulations or injectables or patches or implants, where essentially we can protect people for a longer period of time. So it's not one pill once a day or even episodic dosing, because the data on episodic dosing, even though we're going to go out and collect some, so far hasn't supported that is a sustainable or reliable process for delivery of PrEP. Ultimately, all of these prevention strategies come together in the United States as reinforcing the national HIV/AIDS strategy with the goal of reducing incidence increasing access to care, and optimizing health outcomes. And if you think about test and treat in the context of the National HIV/AIDS strategy, it really is about all three of the objectives of the President's plan. We really need to be able to pull the full component of testing, linkage to care, treatment, and adherence counseling to pull test and treat together so it really does enforce the National HIV/AIDS strategy. Again, coming back to our spectrum of activity, I want to give a word of support to the CDC project, the 12 Cities project that they've organized to really look at how we can work better as the components of the federal government to pull together to really improve the delivery of care, working with the health departments and with the NIH to make sure that we explore the best ways to, in many ways, pull together the resources in cities, the 12 jurisdictions of highest incidence in the United States, to address the challenges of delivery of prevention and treatment within these 12 jurisdictions. Ultimately, each jurisdiction will help develop their own combination prevention product, and implementation will begin in year 2. Again, this is an active collaboration across the federal government, and it really is helping to advance the cause of the National HIV/AIDS strategy. One of the other areas that we have elected to focus at NIH is to see if we can devise a system, primarily working with our partners in Washington, D.C., that will really allow us to create the scientific basis of a strategy that could become a model for how to deal with other U.S. cities with expanding epidemics. And what we have done is worked with the Department of Health in Washington, D.C., and with George Washington University, and there are 2 people at the meeting from GW here today that are actively involved in this project. What we're trying to do is establish effective measures to help control the epidemic in DC, and part of that is then to create a citywide data system that will really allow us to measure the effectiveness of intervention programs. The 4 major pillars are really, can we identify the highest risk populations in a city? Can we seek those people out? Can we find those individuals? Can we get them into care, get them into studies? Again, create this citywide data system that will allow us in real time to monitor the viral load and other parameters. We have augmented the HIV subspecialty care across the city by opening clinics to deal with hepatitis C and other comorbidities. And ultimately, a key other piece of this is the 065 protocol. So as we look at the range of issues associated with seek, test, and treat, or test and treat, there's a range of sort of scientific issues that need to be studied and optimized in order for us to really have an impact on this cascade. Office showed this slide this morning, and it really does capture in real-world data at the domestic U.S. level how the cascade breaks down to where at the end of the day only 26 individuals out of the initial 100 are fully suppressed. And the goal is ultimately, with any seek, test, and treat strategy, is to optimize each of these activities to as close to 100% as possible. With a little bit of a background in organic chemistry, if the yield you routinely get as an organic chemist is 26%, you have no business being in the chemistry business. So as scientists and physicians, we need to really take sort of an organic chemistry approach and get as close to 100% as possible. So again, the HPTN 065 design is, how do we optimize testing? How do we optimize linkage to care? How do we integrate, initiate, and continue ART according to guidelines and maintain viral suppression? And there's a specific randomization at each one of these steps for expanded testing, for continued monitoring of provider and patient knowledge and attitudes toward financial incentives, as well as delivery of antiviral therapy. The linkage to care also involves financial incentives, as does viral suppression. So this is, in many ways, state-of-the-art, cutting-edge approach that as we get the results of this study, how do we improve this or roll this out into other communities? I think there'll be some interesting activities that we can discuss about financial incentives related to possibly adding lotteries to financial incentives or other ways of substitution. I mean, it becomes a little like substitution therapy. You get people addicted to financial incentives. How are you going to substitute something else in to wean them off of their financial incentives? But that should be a problem we'd be happy to have. There are some very unique features about 065 that I wanted to highlight. This really does represent a departure for the NIH in terms of the partnership is not through our traditional NIH-funded sites, but really we're working with community providers and the departments of health in the city to pick essentially clinics and hospitals where testing and treatment is provided. At the same time, it's been a great partnership with the CDC to really get the testing done in some significant ways. It is a multi-component intervention study. Many people have pointed out this fact throughout the day that Seek, Test, and Treat is in fact a multi-component intervention, and ultimately we will have data that comes out in 2014. I'd like to say a word or two about some of these other selected research issues, specifically on the efficacy of ART in preventing transmission and the benefit to the individual. And Wafa highlighted this study this morning, and really 052 may be our only opportunity to really answer this question as the pair simultaneously. Because this is a couple study and a discordant couple study, it will allow us to measure the impact of early ART on disease progression. And it will also allow us to capture data on the efficacy of ART in preventing transmission. In the future, through the START study and other studies, we'll be able to look at the impact of early ART on disease. But I think this may be our only opportunity to really answer this question in a single individual trial. The study began in 2005 and is forecast to go for a little while longer. Again, we'll see how this study progresses, but it is now fully enrolled. The START study also was mentioned this morning, and then again, here too, this really gets to a question of equipoise. Based on the data available, does treatment beginning above 500 have a profound benefit to the individual versus waiting till the CD4 count falls below 350? Enrollment opened in March of 2009. And if there are places that would like to be considered, I imagine that Dr. Neaton at the University of Minnesota would love to talk to you, particularly if you have a significant number of patients that you could contribute to this study. Again, what we're trying to do is balance it in regions around the world. We have elected to cap enrollment in Kenya at about a couple hundred because we do not want to have the predominant set of endpoints coming out of the study being driven by tuberculosis. In terms of across the NIH, the other activities that we are supporting, the first two bullets deal with an international set of studies that are currently being solicited for. So the PEPFAR provided funding to USAID, to NIH, and to CDC to go out and solicit for combination prevention interventions in PEPFAR countries. The CDC request for applications closed April 4th, so all their applications are in. The NIH solicitation was released March 11th, and applications are due midweek next week. And the USAID PEPFAR team elected to support their activities by going through Johns Hopkins University. So again, this will be an area that as the three large federal agencies make these awards, to try to standardize some data elements so that essentially out of the three experiments we can get at least information that can be combined via meta-analysis. Additional NIH opportunities are listed at the bottom of the slide there. For those of you who are involved in NIDA's prisoner population studies, you're well aware that these 12 grants have been awarded. Apparently there was a meeting here that that Jacques and his team got organized that really allowed the team to start pulling together and working in a collaborative way, which is really a great step forward. Additionally, there will be 10 new seek, test, and treat grants that we expect to at the NIH level to be awarded this summer. We have a number of other RFAs and solicitations that have come out or will come out in this space. One of the larger areas that will come out over the next period of time will come from NIMH, where they will be focusing on some of the behavioral aspects around accessing treatment, accessing testing, and linkage to care. So with that, I'll stop and turn it back over to Ed.
Thank you.